Welcome dear viewers to the video on content uniformity in pharmaceutical formulations. So whether you are working on the oral solids or oral liquid or other type of formulations, so content uniformity is critical and very important for the pharmaceutical formulations. Content uniformity is basically critical because it is required that each dose of the formulation or each dose of the medicine should give the proper amount of drug. Proper amount of drug means based on the label claim, it should give the dose in some range. If you think about the assay or potency, then assay is the content of that formulation in composite way, but CU is the assay of each unit or assay of each unit dose you can consider. Each dose of the medicine, each dose of the formulation should contain the amount of medicine in certain range of the label claim, and it is required to be demonstrated that your API is present throughout the batch or throughout the dosage unit in some range or uniformly distributed. It is not like that some units are containing more amount, some units are containing less amount. So content uniformity means the medicine or the drug or the API is distributed uniformly throughout the batch and throughout the dosage units. Now let's start with the video, and in this video you will be able to understand all the things regarding content uniformity. You will be able to understand all the points. You will be able to give answers to all the questions asked on the content uniformity topic. So be with me till the end of video, and I assure you that you will be having very good information about this topic and very good understanding. Content uniformity is also referred as CU in the pharmaceutical industry. It means similar and consistent distribution of API or drug substance or drug in the dosage forms in each unit dose, like tablets, capsules, powders, or the other formulations like suspensions, emulsions, or whatever. Whenever you take the dose of that medicine, you will get the exact amount of drug consistently. CU must be ensured in the pharmaceutical formulations. CU is must to ensure that each unit of the product provides the intended dose to the patient. That means, as per the label claim, the dose should be there. Then, why content uniformity is important and why content uniformity is required? Because content uniformity affects the safety efficacy of the formulation. Why it is affecting the efficacy? Because if content uniformity is not proper, the patient may get low amount of API or high amount of API or dose. So to ensure that each dose provides the same therapeutic effect, same therapeutic action or pharmacological action, CU becomes crucial for drugs with narrow therapeutic index. CU is required for all the type of formulations, all the type of drugs, but it is more crucial for the NTI drugs, that is narrow therapeutic drugs. Safety. Safety is to have the same dose so that the overdosing or underdosing can be prevented. If there is low content, it may lead to action, low action or less action or no action, and leading to treatment failure. And if it is overdose or if it is containing higher amount of API, then it may lead to toxicity. So as per the regulatory standards, regulatory requirements and compliance, CU is required to be demonstrated. So there are mainly two methods for the content uniformity testing, as we know about the weight variation test and the CU test by UV or HPLC. So testing methods for assay is used to determine the content uniformity. Analyze the amount of API in each dosage units using techniques like HPLC or weight variation. Weight variation is done for dosage forms that contain higher amount of API. Now, what are the factors affecting the content uniformity? So, factors are batch size, manufacturing process, and formulation factors. If batch size is much large, there may be chances of CU failure or CU criticality. The size of the production batch can influence the content uniformity. Larger batches may require more rigorous mixing and processing to achieve uniform distribution of the API throughout the batch or throughout the dosage units. Manufacturing process factors like blending time, granulation method, compression, process time, compression machine speed, capsule filling speed, machine speed, capsule filling process time have impact onto the content uniformity. Then formulation factors. The composition of the formulation, excipients, their levels or concentration or proportions or interaction with the API and manufacturing process has great impact onto content uniformity. Percentage of API in the formulation has a very great role in the content uniformity. If API load is low, there are more chances for the criticality in the content uniformity. If the API load is high or medium, then there will be less chance of the content uniformity failure or content uniformity criticality. Regulatory guidelines are there for content uniformity, like USP guidelines are there, USP monographs are there, general chapters are there, and ICH guidelines are there. Also, ASTM guidelines are there for content uniformity. So, what are the strategies to ensure content uniformity? First is formulation optimization, process optimization, selecting the excipients and their optimization, their ratios to be checked, and its effect onto the content uniformity is required to be studied. Use of advanced mixing techniques like high shear mixers, other technologies to enhance blending uniformity, like use of the blenders, use of some intensifier bars in the blenders, regular quality control testing, performing routine CU testing during manufacturing process to identify the process problems and inconsistencies. Process validation is must, and validating the manufacturing process is required to ensure consistent results over time. Proper blending time to be done, proper blender RPM to be selected, occupancies to be evaluated. Nowadays, NIR techniques, PAT techniques, and PAT tools are being used for determining and ensuring the content uniformity and batch to batch uniformity, and to avoid the batch to batch variation in blend uniformity and the content uniformity. So now for content uniformity, AV value is terminology, main terminology, and based on that AV, we can say that the content uniformity test is passing or failing. So acceptance value is a statistical parameter used in the content uniformity testing to ensure that each dosage unit contains the same amount of medicine or drug. And AV value gives you idea about the content uniformity is within the acceptable range or not. It helps to determine whether the batch meets regulatory specifications or uniformity. AV value helps to ensure batch consistency and is especially important for narrow therapeutic index drugs. For these drugs, the precise dosing is crucial to avoid therapeutic failure or toxicity. As per the USP, specific criteria is there for evaluating the content uniformity. So content uniformity is done by CU test and also by the weight variation testing. Content uniformity criteria. CU testing is required for all dosage form with low dose active ingredients. And when you can say that the CU testing by HPLC or U is required, and what is the definition for low dose formulations? So USP gives idea about this. If the formulation contains less than 25 mg of the API, or if the API is less than 25% of the tablet weight or capsule fill weight, then CU testing is required. And if the formulation contains the API more than 25 mg or above the 25% of the tablet weight, then you can go for the weight variation testing. Test method involves analyzing the 10 individual dosage units for assay and then calculate the AV value. AV value limit is required to be equal to or less than 15 for the 10 units testing. And if this 10 units testing fails, then you can go for the another 10 units, another 20 units, first 10 units, then another 20 units, so total will be 30. And for 30 units, AV value is required to be between 15.1 and 25. For 30 units, the mean content must be within 85% to 115% of the label claim, and no unit should deviate more than 25%. So this is the criteria. First test is on 10 units and second test is on 20 units. So total units will be 30, and based on that AV value criteria is there. So for first 10 units, the stage is L1, and then another 20 units, the stage is L2. Now coming to weight variation testing. Weight variation is used for formulations containing higher doses or high API percentage, typically greater than 25 mg or more than 25% of the tablet weight or capsule fill weight. Test method. Weigh 10 dosage units individually. Calculate the mean weight and determine the deviation of each unit from this mean. Weight variation criteria is given in the USP. If tablet weight is less than 130 mg, the deviation is 10% plus minus 10%. If the weight is between 130 to 324 mg, the deviation is 7.5% allowed. For the formulations or tablets or capsule fill weight is more than 324 mg, 5% deviation is allowed. So for tablets it will be the tablet weight, and for capsules it will be the fill weight. Now coming to the AV calculation formula. So AV calculation formula is given here. AV is equal to m minus x plus ks is the formula. Here X is the mean of 10 tablet results. M M is calculated or determined based on to the X. If X is between 98.5 to 101.5, then M is equal to X. If X is less than 98.5, then M is equal to 98.5. If X value is greater than 101.5, then M will be equal to 101.5. K is the acceptability constant for 10 tablets. Its value is 2.4, and for 30 tablets its value will be S is the sample standard deviation, and based on this criteria the AV value is calculated. AV limit is there for L1 and L stage. L1 stage AV value not more than 15 for 10 tablets, and for L2 stage AV value is not more than 25. So this is regarding the AV value calculation. See, the content uniformity is related to the blend assay and blend uniformity. These are interrelated, and that's why blend assay and blend uniformity is required to be tested before compression and capsule filling for oral solid formulations. And blend assay and blend uniformity will give you some confidence and idea about the content uniformity. So this is regarding the information on content uniformity for pharmaceutical formulations. I will make upcoming videos on the relation between the content uniformity and the stratified sampling, so that you will have idea about the differences and similarities and interrelation between the stratified sampling and the content uniformity. But for now, please remember that stratified sampling is to have the confidence onto the CU, and CU testing is the finished product testing. Stratified sampling is required for validation to demonstrate that the process is capable to get the CU within range, while CU is the finished product testing. I will make separate video for the relation between stratified sampling and content uniformity. But for now, you understand that the content uniformity testing is finished product testing. Blend uniformity, blend assay affects the content uniformity, and content uniformity is critical to achieve for the low dose formulations, like some hormone formulations are there, some vitamin formulations are there, then some formulations contain very low amount of drug for therapeutic drugs. CU is very critical. There are different ways to achieve the content uniformity. Sometimes the process modifications are done. Sometimes the process optimization are done. Sometimes manufacturing processes are changed. Sometimes the tablet weight or capsule pill weight is required to be optimized. So there are many ways to get the content uniformity within a range. Content uniformity is must to be demonstrated. Otherwise, if content uniformity is failing, the product application may get rejected, or you will get many more queries. As content uniformity affects the safety and efficacy, it is required to be demonstrated through the process development and validation batches. Content uniformity testing is done on all the batches which are manufactured for commercial marketing, and without passing the CU test, the batch cannot be released into the market. So I hope I have covered some of the critical informative points. and I hope this video will help you to have answer to many of the questions. So thank you for watching the video, and keep watching those videos to have good understanding about the topics. Thank you.